So I'm starting the recording. So welcome everyone for the DSBS seminar series. It's the first seminar of our series we wanted to start with you this year. So this follow the requests we had from many of you during the last symposium at the DSBS symposium in France, during the online event where many people were saying they wanted more opportunity to share their work and interact with the society more regularly. So we hope you will enjoy this opportunity. We're trying to start. It's a test, so don't hesitate to give us any feedback after or at the end of this meeting. So today we will have two presenters that will talk about the biogeography and the taxonomy of species from chemosynthetic ecosystem. So we have, sorry if I misspell, Guilherme Siqueira Toledo de Carvalho from the University of Sao Paulo that will talk about the wood-eating bivalve from the Southwestern Atlantic, and he will present first his talk. And then we will have Melissa G. Betters from Temple University that we talk about the evidence of depth partitioning, the Provanna gastropod that she found on the Costa Rica Margin. So Guilherme, you can start your talk.
Okay, let me just share my screen now.
Yeah.
Can you guys see it?
Yeah,
okay.
I can see it.
Is my like your guys video showing on the right? Because it's showing for me. I don't know if it's covering something for you guys, or do you see just the presentation?
I can see you and the presentation. I think okay. It looks good apparently for the viewers.
Let me just okay. So hi everyone, thank you for being here. As Pierre already said, I am Guilherme. I am a master's student at the Instituto Oceanográfico in São Paulo, Brazil, at the University of São Paulo. My advisors are Paulo Sumida, also here from the Oceanographic Institute, and Flavio Passos from Unicamp. And today I'll talk about the wood-eating bivalves of the deep Southwestern Atlantic. So first of all, why talk about wood in the deep sea? Usually people don't really think about wood when they think about the deep sea, right? It's a very alien subject in our area. And to quickly illustrate it, let's take a trip to the antipodal point of where I'm talking right now. So let's more specifically the Southeast Asia. Here you can see mainland China and this relatively large island, Taiwan. Here in 2009, huge typhoon, the Morakot, and later renamed the Zetsani typhoon, hit the island. And it was a very catastrophic event. You can see here in the picture a river from Taiwan, where you can even see some bits of the landslide here in the back. Let me just put a laser pointer. Here you go. You can even see a landslide in the back, and the river is full of wood debris, right? But how much wood does actually reach into the ocean? It's not a question that we can actually answer right now. But on this single event, around 3.8 to 8.4 million metric tons of wood were transported to the oceans in one single event. And this is in a relatively large island, but still it's an island. So one can only imagine all the impact from rainfalls and landslides and typhoons from around the world in a given year. But how does that wood actually reach the ocean, right? Wood first, after landslides and rainfalls and etc., it gets carried to the ocean via rivers. Then it floats for a while as driftwood, saturation with seawater, and after that it sinks. Then two things can happen at once. Either it gets consumed and then gets uptaken into a vast gamma of deep sea food webs, or it could part of it could enrich the sediment and end up sequestrating some carbon. And one of the main taxa involved in actually degrading the wood at deep sea are bivalves. The main group in the deep sea are the Xylophagaidae. And to illustrate it, here is an individual showing the general morphology of the group. It has a more or so typical bivalve body, except this opening on the anterior part of the animal, but we'll enter in more details a little bit later. So who are the Xylophagaidae? They are in the superfamily Pholadoidea, which according to this molecular study, which is the most widely accepted today, we have at the base the Pholididae, also known as Pidoc or angel wings. And as a sister group to the Xylophagaidae, we have the Pteridinidae. The Pteridinidae, I think, are the most commonly group of the superfamily, which are also known as shipworms. They have a worm-like body, which is very unusual for morphology in bivalves. They tend to be present in any driftwood that you can find in beaches, or even are known to do lots of man-made, to destroy a lot of man-made structures. But they are mostly restricted to shallow waters, reaching at most 200 meters deep. Then there are the Xylophagaidae, which we just mentioned, and they are mostly restricted to the deep sea, with one species reaching up to 6,000 meters deep. But they have been found in intertidal waters. But there's some discussions in the validity of those records. Regarding how they eat the wood, or in other words, their xylotrophy, here on the left, you can see one of their piece of wood with some of their burrows. It's each one of these little holes here. You can see some are occupied and some are empty. And here on the right, you can see one of them actually occupying one of those burrows. And I really like this picture because I think it gives a pretty nice idea on how they actually dig their burrows. Here on the interior part of the animal, you can see these white streaks here at the bottom. And when you zoom them a bit, you can see they are made of little denticles. They actually use to rasp the wood with lateral movements of their shells. And so the objectives of my study as a master's student was to assess the Southwestern Atlantic composition of this family. To do that, I first identified any different morphotypes I found. Then I will formally describe any new species or genera, if any at all. And I would like to examine any differences in their morphology or ecology, given their differences in substrate or in depth. So, second. So to the actual field work. Here you can see the South America. And more specifically, our work was focused on the southwestern coastal areas of Brazil. If we zoom in a little bit, you can see some points that our laboratory did on a previous study on SP. SP stands for São Paulo, RJ for Rio de Janeiro, and ES for Espírito Santo, which are states in Brazil. And next to it are the depths, 1,500 meters or 3,300 meters. At those points, they found the spiny apex genera of Xylophagaidae, the Xyloredo at these 2 points, and Xylophaga at this point. So to do a comparative study, we put a lander, which is an autonomous structure, which I'll show in a little bit. We put it a little bit shallower at 550 meters here on the white circle. And it's a point 240 kilometers off the coast of Santa Catarina, Brazil. It stayed submerged for 10 months. And as I said, 550 meters deep. The actual lander structure is pictured here. And how it works is you can submerge this structure and with buoys and an acoustic releaser. And what you see here, these baskets, they are made of a net, which can pass water, but only small organisms. And then in those nets, you can put different materials. You can have wood, you can have different types of wood, you can have whale bones, and you can even put a control substrate, such as a basalt block or concrete block or whatever you feel like. Here is a picture just to quickly illustrate the blocks on the field. You can see that there were 3 wood species of 2 different sizes. There were even some coconuts, and here on the left you can see the whale bones and cow bones. Here you can see the material in the actual study area at 550 meters. And this picture was made possible by the deep rover submersible of the Alucia, MV Alucia, manned by a pilot. And on the left, you can see my advisor Paulo. So to the actual results of our work, in around 400 cubic centimeters of wood, which is not a bit much than what you can hold in both of your hands, we found around 1,700 Xylophagaids of 3 different genera, one of which is a probable new genus. Their relative distributions were as follows. You can see some differences. The colors represent each of the species we found. And on the left, we have a higher density wood. I think the direct translation would be pink cedar. And on the right, you can see a soft wood of lower density wood. We call it cacheta. It's a wood made to make boxes and stuff like that. Not pictured here are the Xylophagaidae found in an even higher density wood, Garapera, because we found only around less than 10 Xylophagaidae, which were too small to even be able to identify. So to quickly illustrate it, who are those 3 genera? Here's the Xylonara Atlantic, which I already show you, a general morphology of the genera. They have a mesoplax, which is an accessory shell that they have, which covers their anterior adductor muscles, probably to protect it from abrasion against the wood. And you can see a SAM image of their siphon tips. And pay attention that their siphon tips is nude. It has no attached structures. Here we have a member of the Xylophilas genus, and you can see there are a few key differences. One of them is the thick periostracum on their siphons and the presence of 2 periostracal plates near the siphon tips. Same image of the plates reveal they have a very unusual ornamentation near the siphon tips and series of periostracum shifts near the tip, almost leaving it closed. And then there's the probable new genus, which we will leave as Xylophagaidae species 1 for now. since it's not described yet. And it has a very unusual morphology for the group, which are these calcareous siphonoplates here at the tip of the siphon. Here you can, on the left, on the right, you can see them. It's 4 plates actually, and it's 2 dorsal plates and 2 ventral plates. And they bear some resemblance to calcareous plates also found in their sister group, Pteridinidae, especially when you look up close, but they have some key differences, especially the amount of plates. In the Xylophagaidae, as already said, it's 4 plates. In the Pteridinidae, it's just 2. And also some differences in muscle and how they insert in the muscle and stuff like that. So it's a little bit early to make any evolutionary relations. But the plates on this probable new genus, you can see these little spines here on the distal part of the siphon tips, which when we do a zoom and images of electronic micrography, you can see those spines point anteriorly, which is probably, I think at least, it's an adaptation to help close their burrows. And in dealing with Xylophagaidae, a very powerful tool are micro CT scans. You can do both of the wood blocks. Here on the left, you can see a wood block, the external surface of it. This is just a rope that we use to keep attached the block identification. And using this X-ray tomography, you can see the actual bivalves inside. Each little gray dot here is a Xylophagaidae. You can see some bigger ones and some smaller ones. And you can even, changing the density of your CT scan, you can even see their burrows. I didn't put them all here because it would be a mess of an image, but you can then estimate their growth rates and the composition rates of the wood. Here's just a video to quickly illustrate that. It's also 3D, so you can estimate their relative density, their relative position in the wood, and you can even do a CT scan of the organism itself to assess some morphological features of their valves, and not pictured here, but you can also assess some internal anatomy characteristics using the micro CT. So to conclude, first, we have new reports for 3 genera in the Southwestern Atlantic: the Xylonoras, Xylophilus, and this probable new genus, which shows that it's probably a vastly understood area and in need of more assessment, since just one lander brought up these 3 new genera. One can only imagine what a full study of the coastal area of Brazil could entail. So we have a high diversity of genera, which is possibly related to the vast forests along the Brazilian coasts. And this diversity could be endangered by the high deforestation rates that we see on the last centuries. And a new morphological characteristic in the Xylophagaidae, which are the calcareous siphonoplates that I just showed. And some future perspectives for my work. And we need more populational data, especially more wood species and more South Atlantic locations, not only on the South America, but we also need a lot of studies on the Atlantic African coast. We need more macromolecular sequencing of some species, especially barcoding and some mitochondrial ribosome genes. And I want to do a call out here to a very important subject, which is to study the relationship of the Xylophagaidae, the composition of wood, to the global carbonic cycle. because we have no estimates, no good estimates about how much wood enters the ocean, how much of that wood gets uptaken, how much of that wood ends up in the sediment, and how that impacts global systems of CO2 and global warming in general. So I just want to thank first everyone for being here and my institution for allowing my work. I want to thank the society for presenting this opportunity to present my work to everyone, especially the mentoring group, which helps me every month on our meetings. I want to thank Marcel Sayago, one of my international collaborators, the OceanX and their crew at MV Alucia, and my funding agencies. Here you can see a picture of me and my advisors, Flavio and Paulo Sumida, and my contact info if you want to reach out. Thank you very much.
Thank you, Guilherme. It was really nice talk. Really like the CT scan and data. Really nice. So we have time for a few questions. So does anyone have a question? You can raise it on the chat or raise his hand and talk directly maybe. Yeah, if no one has a question yet, I have one for you. Well, Frank, do you want to start? Yeah, go ahead.
Oh yeah, thank you. Thank you. So these animals have parasites, and would they arrange like this little teeth you show? Do they have parasites? Sorry.
What's the question? Sorry.
I wonder if these gastropods have parasites.
If they have any parasites.
Yeah. Did you ever find any parasites on these guys?
I didn't find any parasites, especially since they're very small, right? The general I found, they are, I think, in the 1 millimeter length of their shell. So I still didn't do any internal anatomy, but we did find some polychaetes, probably predating the Xenophilidae. Usually we find empty shells full of some polychaetes, and sometimes I have even found a polychaete actually inside going in one side and exiting on the other side of the Xenophilidae. So not actually parasites, but we did find some predators.
Thank you.
No problem.
Another question from someone else?
I have a question.
Yeah, go ahead.
So with the 3D CT scans, because you had the way that the lander was arranged in kind of like a 360-degree arrangement, has there been any talk about looking at like directionality of where these bivalves are like coming from?
Yeah, if you in the picture I showed about the lander, you can see that we had some blocks inside the actual baskets and some on the lid of the baskets, and not a directionality, but we were trying to assess if ocean currents would have an effect, right? Because the ones near the bottom would have a slower current, and the ones near the lid would have stronger currents. So not really the directionality, but we're trying to assess if we can find any differences in oxygenation or position relative to the bottom.
Thanks. There is another question on the chat from François Lallier that is asking, are you planning to look after the symbiotic bacteria that sometimes help this guy digesting the food? And I will add one question related to this one. Do you think there could be some, because like you see the same species, like the 3 species on the same block, right? So is there some niche partitioning in terms of food, like the way they degrade the wood? Are you expecting different nutrition, different specialization in the way they eating the wood between the species? Or do you think they have like the same overall regime?
Okay, first answering the symbiotic bacteria. I would really like to, but I'm not a microbiologist, but I think there's a much needed work on their symbionts. We have, for example, there is a big mystery on how they actually digest lignin. There has been some great works trying to solve that issue, but we still don't have many clues on how their symbionts, or even if the bivalves themselves can actually digest the lignin. The cellulose, they have some enzymes, but the lignin itself, not really. And answering the other question, and yeah, and being sincere, I have no idea if there's niche partitioning in the different species, because to actually assess that, you have to do some isotope studies, right? I think it's a great approach, and there has been some work done on them, on other species, and you can see there is some gradients of how if they are mostly xylotrophic, if they are mixotrophic, or they rely on plankton probably. So without any real analysis, it's really hard to say.
Okay, cool. And also about I will leave the question on the chat after, but the CT scan, can you distinguish the three species on the CT scan? And do you see some spatial distribution, like different spatial distribution between the species?
The one I put in the slides specifically, you can see some features on their shell, because they are, at least in the blocks that we analyzed, they were the only one which had the mesoplex, which was the accessory shell on the anterior part, and you can see them pretty clearly on the CT scan. But the other two, they don't have any distinguishing features. Their mesoplex is just a periostrical sheath covering their anterior part, and the very small individuals, you wouldn't get a good definition of morphological features. So you can, but not all the time.
Yeah, just for the big one. Okay, right. Just one last question, maybe from the chat from David Barros. Do you think the build of dam in the river could significantly affect the total amount of wood reaching the sea, and I guess affect the distribution of your wood bivalve?
Sorry, can you repeat it?
The dam, all the dams that are built on the river, are they affecting the amount of wood that are reaching the sea in the end, and the potential of surface?
Yeah, there's a lot of potential in research dealing with this interface between riverine studies and ocean studies, right? People studying inland rivers and lakes there's a lot of work dealing with how wood impacts those habitats, but not so much when we go to the ocean. There's not a lot of studies dealing on how that impacts the availability of wood in the ocean.
Okay. Okay, thanks. I think we're gonna go for the next talk from Melissa about the Provanna gastropod on the Costa Rica margin. If there are more questions on Guilherme talk, we'll take some time also at the end to like interact and continue the discussion. Melissa, you can start.
Okay. All right, let me just share my screen here. All right. Okay, so my name is Melissa. I am a PhD student at Temple University. I work with Eric Cordes. This is research that we've done on Provanna snails from the Costa Rica margin, also in collaboration with Abigail Keller. All right, so to begin, this research mainly focuses on species range limits. So a species range limit is essentially an observable expression of a species ecology and life history. So for instance, if you have a species of snail that lives at the bottom of the ocean, if you give that snail sufficient gradual enough environmental gradients, sufficient enough genetic variation, balanced dispersal, as well as time, that species could theoretically adapt and expand to live anywhere. However, as we know with most organisms, that type of cosmopolitan distribution is relatively rare. So instead, you tend to have these abiotic and biotic factors that limit a species' range. So for instance, you could have abiotic factors such as steep environmental gradients and barriers to dispersal that limit a species' range, as well as biotic factors such as competitor species and predators, which further limit a range. And these biotic and abiotic factors tend to interact in predictable ways. And one of the patterns that we know of in which they interact is known as the species interaction abiotic stress hypothesis. So essentially, if you plot a species distribution across a stress gradient, in high stress environments, abiotic conditions tend to exert more influence on a species range, whereas in low stress environments, you tend to have biotic factors exerting more influence on a species range. So with this all in mind, I approached our investigations into the hydrocarbon seeps at the Costa Rica margin. And so we sampled 6 hydrocarbon seeps across 3 research cruises from 2017 to 2019. And the organisms of interest in my study are the genus Provanna, which is a genus of deep sea snails, of which we collected over 1,800 specimens. And so my research aims for this study were to first identify the species of Provanna found at the Costa Rica margin, and then to characterize the distribution of these species, as well as determining the potential biotic and abiotic factors responsible for those distributions. And so to conduct this research, we used a bunch of different tools. Mainly, we used the ROV Falcor and Atlantis to conduct our research cruises. We also utilized the ROV Sebastian, the AUV Sentry, and the HOV Alvin to conduct biological and environmental sampling. And so my first order of business upon getting all of these Provanna specimens was to characterize them into different morphotypes. And so it turned out we collected 4 distinct morphotypes from the Costa Rica margin, the most abundant of which was tentatively described as Provanna laevis, morphotype 1, which had over 1,600 specimens collected. The next most abundant was tentatively described as Provanna goniata, with 180 specimens. And then the last 2 seemed to be more opportunistic samples. Provanna pacifica and Provanna lomana both had less than 10 individuals sampled across all 3 years. And so to verify the identity of these species, I conducted barcoding for the mitochondrial CO1 gene, which confirmed morphotype 1 as being within a single molecular taxonomic unit of Provanna laevis, as well as Provanna glabra from the Western Pacific. Morphotype 2 was grouped together with Provanna ios, which was a novel result. The morphotype of Provanna goniata prior to this study had never been barcoded before. So this affinity to Provanna ios was relatively unexpected. And then finally, morphotype 3 is most likely going to be Provanna pacifica, given its morphology as well as location. However, similar to Provanna goniata, Provanna pacifica had also never been barcoded prior to this study. And so once I verified the identity of these species, I began to examine where they occur. So for the purposes of this study, 2 of the 6 sites, Capo Seep and Mound 11, had relatively low abundances of Provanna, 5 and 6 individuals respectively. And so these sites were excluded from analyses because these, in our mind, did not represent permanent populations of these organisms. So instead, our research focuses on Provanna laevis, which occurred across 3 of the sites, and again, very high abundance, as well as Provanna ios, which was only sampled from Hako scar. And what was interesting about this distribution is that despite the fact that Hako scar and Hako summit occur only 8 kilometers apart from each other, these species never co-occurred. And so I became interested in why that might be. So my initial thought was that it could potentially be the difference in chemosynthetic context, particularly at Hako scar. So Hako scar is what's considered a hydrothermal seep, in that it has certain characteristics of hydrocarbon seeps as well as hydrothermal vents. In particular, these thermal anomalies at point sources of fluid, sometimes up to 9 degrees C. And you can see in this video Provannidae living very near these thermal anomalies. And so that was my initial thought. However, a lot of research agrees that differences between vent and seep fluid rarely is sufficient to filter species. And indeed, Provanna laevis and Provanna glabra, that single molecular taxonomic unit, is found to occur at both vents and seeps, with very little genetic differentiation across all bar-coded individuals. There's only a maximum pairwise sequence divergence of 2.24%. And so instead, research points to a lack of suitable substrate or oceanographic conditions associated with depth as potentially more influential in filtering species between these environments. And so I chose to look at that in a bit more detail. So first, looking at suitable substrate. So first, all of our Provanna came from one of 4 substrates primarily: Bathymodial mussels, authigenic carbonate rock, fallen or deployed wood, as well as siblaglinid tube worms. And the distribution of these species broke down like this. And what's notable here is that for Provanna laevis and Provanna ios, they both occurred primarily on mussels and tube worms. And actually the differences that you see here in their distributions mainly has to do with the differences in the distribution of those foundational taxa, rather than any express preference of these Provanna species. So as far as lack of suitable substrate, that didn't seem to be the case for this species filtering. So then I looked at oceanographic conditions associated with depth. And the Costa Rica margin occurs in the tropics, which means that there are steep environmental gradients that tend to occur in the upper 2,000 meters of water, which you can see here exemplified by oxygen, temperature, and salinity. And these data were collected via CTD casts deployed from our research cruises. And so if you compare the mean depth at which Provanna ios occurs, which is around 2,000 meters, to that of Provanna laevis, which was around 900, 1,000 meters, you can see that they encounter very different environmental conditions, particularly when it comes to oxygen. And examining the data in detail, the mean environmental conditions that they encounter were significantly different across all 3 of these parameters. And particularly, again, for Provanna laevis, it was found to live very near the oxygen minimum zone at this site, exemplified by this purple band here of very low oxygen conditions, sometimes being near anoxic. And so to visualize this a different way, I conducted a principal components analysis examining the factors associated with depth, as well as oxidative reductive potential, which was used in the study as a proxy for seep activity. The lower the ORP values, the nearer to the point sources of fluid the specimen tended to be. And so what this PCA plot indicated to me was that it was the factors associated with depth rather than seepage that seemed mainly responsible for parsing the 2 species. And of particular note here is that while Provanna laevis inhabited a larger range of ORP values than Provanna ios, their ranges did overlap. And so I began to wonder whether one species was potentially excluding the other, and if that's the reason why they don't co-occur. So I investigated this using the principle that ecologically similar species should theoretically react to environmental stressors in comparable ways. And I examined these ecological reactions by looking at their shell morphological traits. So examining a plethora of morphological traits in reaction to the environment, I found that Provanna laevis at the Costa Rica margin tended to be larger at both greater oxygen and lower temperature values, indicating that their growth was positively influenced by deeper water conditions. For Provanna ios, they were found to be less spiky, had less textured shells, at both lower salinities and higher temperatures. So something about their shell calcification seemed to be somewhat inhibited by the conditions of shallower water. So what this told me was that while Provanna laevis seemed to prefer greater depths, it seemed that so did Provanna ios. And so all of this together sets up evidence for competition for available substrates, for seep fluids, and for living at those deeper sites. And so when two species compete with each other, there's always going to be a winner, there's going to be a loser. There's the range of a superior competitor tends to be set by the abiotic environment, whereas the range of an inferior competitor is often set by the superior competitor, and in that it's cordoned off to areas not physiologically tolerable to that superior competitor. And so looking at the biogeography of these species as a whole, the known range of Provanna ios and Goniata, again, one molecular taxonomic unit, is relatively small. It spans 1,000 meters depth, tends to be below those steep environmental gradients in the upper layers of the water, and only occurs in a very thin band of latitudes and longitudes. Meanwhile, the known range of Provanna laevis and glabra spans over 3,000 meters depth, including those steep upper gradients in the upper layers of the ocean, as well as pretty much the entire northern hemisphere of the Pacific Ocean. And so these known biogeographical distributions tells me something interesting about the different evolutionary trajectories of these species, in that it's possible that while Provanna ios may have evolved to be able to outcompete sister taxa in its narrow preferred range, Provanna laevis seems to have evolved the ability to simply avoid that competition, to be able to live in those areas not physiologically tolerable to superior competitor species. And so if you look at the range of Provanna laevis specifically at the Costa Rica margin, it follows very closely the species interaction abiotic stress hypothesis, in that if you plot the stress gradient as depth, those high stress, shallower waters the abiotic conditions tend to limit the range of Provanna laevis here, particularly oxygen concentrations. Whereas in the low stress, deep waters, their distribution seems to be constrained mainly by competition with this sister taxa. And so this study represents a way that taxonomy and biogeography can kind of come together to tell us something interesting about the evolutionary history of these species. And so with that, that's all I have as far as my research. I want to thank the SBS for hosting this talk, for everybody listening, and for everybody who's helped me with this research. And I welcome any thoughts or questions.
Thanks a lot, Melissa. It was a really great talk. Really enjoyed. Remind me a lot from work I did on my model species too. Does anyone have a question for Melissa? Waiting just a few. François Lallier have a question too.
Yes, again.
Oh, yeah, you can go ahead, François.
Yes, it was very interesting to see the oxygen preferenda of the two species, and might be a good idea to look at the respiratory traits, gill size or gill surface, and possibly respiratory pigments.
Yeah, I agree. That would be cool. Yeah, I haven't considered doing that yet, but that would definitely be a very logical next step with this type of research, for sure.
Do you have preserved samples?
Yes, they are ethanol preserved. So I think we have some frozen samples, so might be able to do some of that work. Yeah.
Good.
Frank, I have a question. Are they traced in the fossil record that could indicate change in overlap along the depth range? for this species?
That's a good question. I don't know how extensive. So Provanna laevis, I know, is known from the fossil record, but because its known extant range is so large, the fossil record is not necessarily, doesn't indicate any differences from what we observe today. I don't know of any fossil records of Provanna ios, the other species. But that would be really fascinating because, for instance, we don't know what the lower range limit of that species is. We can only, extrapolate what the upper range limit is in these type of contexts. But yeah, if there are fossil records of it lower, that would be kind of vital information that we're currently missing.
Okay, thanks. I had a question too, like this competitive exclusion made me think about settlement and recruitment of these species. So I don't know if you have looked at the size of the Provanna you had. Maybe it's like a very long work to do. But yeah, so is there do you think you only got like adult, or maybe did you add some settlers? And about the settlers, I was wondering if you could like do some, if you were doing some settlement block, some artificial one on the field, would you expect on some sites to see settlers from both species on the deeper side because of the extensive range of both species, and then a competitive exclusion that would happen after the recruitment? Or do you think this competition occurs after or before in the life cycle?
Yeah, I've thought about this a lot. So one thing that I've had to kind of cut from this talk just for time, there's a whole morphology kind of component to this research that I unfortunately just didn't have time to talk about. For Provanna laevis, for Provanna laevis and Provanna ios, I got a wide range of sizes, both juveniles and adults for both species. So their sizes actually didn't significantly differ from each other at all. So it appears that it's adult, mature populations at both sites. We did deploy wood at these sites from one year to the next. However, even on the wood deployments, there were only settlers of, the respective species. They didn't seem to cross. So one thing that I'm really interested in, and I plan to address this using some population genetics studies later in my dissertation, is to see whether there is directional exchange of individuals between Hakoscar and the shallower sites. Because yeah, at this point, I'm not sure exactly when in their life cycle this is occurring, this filtering is occurring, but that's something that I do want to address.
Because maybe it's a past competitive exclusion that already happened, and now their life cycle is already adapted, kind of.
Yeah.
And the larvae don't come, maybe already.
Yeah, the reason why I think that might, that may not be the case is only because Provanna laevis has exhibited the ability to live deeper, in that it's found, far deeper than where it's found at the Costa Rica margin in other areas of the Pacific. So it seems like it should be able to occur at Hakoscar and yet somehow doesn't.
Yeah. Right. I think there is several other questions. Let me check. Frank is asking, Provanna ios necessarily co-occur with laevis, or do Provanna ios alone at times, and then does it build large population?
Yeah, so Provanna ios was thought to only occur actually on the East Pacific rise. It was thought to be a vent endemic species before it was found here with very little genetic distinction. Right, the main difference between Provanna ios and the morphotype Provanna goniata is its ornamentation. Yeah, it is found alone at the East Pacific Rise, where it does, yeah, exhibit comparable populations to what's seen here.
Okay, thanks. Just before we continue on the question, I have just a few announcements to make before, because I think some people want to leave maybe already. So the second seminar will happen next month at the end of June. We will post again on it and send the Zoom link as we did slightly before the day. There will be more seminar that will happen this fall. I'm going to start organizing them. If any of you or someone in your lab want to participate in this talk, they can contact me or the other trustee, and I will organize with you and set a date. The seminar is a priority for students and early career, but it's open for anyone. So if our more senior scientists in our society want to present their work, they are most welcome to do it. One more thing that might interest young research, so early career and PhD students, there is a seminar organized by the Hong Kong team. You have all the detail on the GSBS website, as well as on Slack. I think we post it regularly. They are doing this symposium on marine science, and there is a 3 sessions. One is fully dedicated to deep sea biology, and they are looking for abstract. They extended the deadline until the end of this month. So if you want to join, the symposium is fully online, it's free for registration, and it's just like posting your abstract, and they will let you know if you have a talk or not. So I encourage people to join. I think it's a nice opportunity for presenting your work too. And there won't be a seminar during December, so it's an opportunity at that time. Yeah, so I'm stopping with that. We can continue with the question. I think Maeva Perez has one. She was asking which of the 2 species colonized this site first. Like, do you know that?
I'm muted, sorry.
Oh, sorry.
I don't think we know that. Yeah, I don't know the answer to that question. I don't know which colonized first. Yeah, I don't know. That'd be, I wish I knew.
Yeah.
All I know is that the wood deployments that we did had colonizers at both of the sites. So there seems to be a ready supply of larvae to settle at these sites for both species.
Yeah. And how far the wood were deployed from the existing assemblage of Provanna?
Sorry, can you repeat that?
Yeah, I'm thinking like so you deployed wood. How far were they in distance to the existing assemblage of the Provanna you found on the site, the natural one?
They were within the same. They were within like the border of the seep site. They were a little bit outside of the like. So we kind of they were in the more of the like transition zone. So they weren't like right in the main dense assemblage of the species. They tended to be more in the transition zone out to the outer regions of the seep. So a little bit lower, for instance, like ORP readings than those in the tube worm bush or the mussel bed.
Yeah, because maybe the problem is also self-recruitment from the
absolutely
existing population.
Mm-hmm.
Frank is asking, is there any growth rate difference? Or do you know that?
Yeah, I don't know
different
that. We did not keep any of them alive. We preserved all of the specimens that we had, so we haven't tried culturing them. I know that Sean Ariano has kept Provanna alive in culture for several years. So it is possible to do. We just did not do them for these specimens from the Costa Rica margin. So I don't know if there's a difference in their growth rates.
Thank you. I saw that I missed one question earlier from Jorge Cortes. Is seeing that the ornamentation of the different species is very huge, and is wondering why. Do you have any idea?
Yeah, so there is some indication that so again, I didn't get a chance to touch on this just because of time, that the spikiness of the texturing of Provannidae seems to be related to proximity to the seep fluid. So the closer it is to the seep fluid, the less textured the shell tends to be. And so, which would explain the difference in ornamentation, for instance, between the East Pacific rise morphotype and this morphotype at the seep, and that these tend to have lower ORP readings at the hydrocarbon seeps. Yeah, I'm not entirely sure what the purpose of the spikes are. My thought is that it could potentially be one of the competitive advantages of Provannidae. If predation comes into play at this site, at Hako Scar, perhaps the spikes, give it a competitive advantage to avoid predation. But I am not entirely sure what the actual function of those spikes are, what the purpose of them explicitly is.
Okay, thank you. I don't know if there is more question. If you want, you can also ask them on Slack. I created the dedicated channel for the GSBS seminar series that you can join. So yeah, feel free to ask any question anytime there, or feedback on the organization of it if you have like complaint or anything.
Thanks, everyone. Bye.
Yeah, I don't see any more question. I think we can finish with this now. Thanks a lot, Melissa and Guilherme, for starting this seminar series. We will, so as I said, the next one is in June on a different topic, and yeah, I hope you will enjoy it. Thanks a lot for coming to everyone, and see you next time.